Hello BSCFT. Today's topic is post harvest diseases. Post harvest diseases are studied under the plant pathology, just the science of plant pathology. Plant disease is defined as continuous malfunction caused by infection by a parasitic microbe. So, whatever the microbe which is living on a plant, fruit, or any part of it, which causes malfunction due to infection. Infection means what? The microbe comes to that plant and multiplies there, utilizing the nutrient and the system of that particular fruit or plant. Continuous malfunction and infection. Malfunction means the normal system working doesn't work properly. So malfunction is caused by the infection into the plant or fruits and vegetables. So continuous malfunction and infection by a microbe differentiate post harvest diseases from defects related to the physical injury, growth abnormalities, injuries caused by harmful environment such as chilling, hypoxia, ethylene insensitivity, sun exposure, etc. Now, what is the point? The point is that the infection or disease is different from the defects, and defects can be caused by physical injuries as well as mechanical injuries or growth abnormalities due to the nutrient deficiency or the sun deficiency or the water or mineral deficiency. Injuries caused by harmful environment like chilling injury is the injury caused by refrigeration, then hypoxia because of availability of low oxygen. Whatever injury is caused to the fruit or vegetable, that will come under defect. Then ethylene insensitivity, that means the higher concentration of ethylene than needed, or lower concentration of ethylene than needed. Then sun exposure can leach out moisture, so it can create defect. Post harvest diseases are those that appear and develop after harvest. Now, outcomes of post harvest diseases. Products with defects are unmarketable, leading to increased cost of production and occasionally loss of an entire crop. Defects because of diseases can cause economic loss. Individual defective fruit require additional handling, hand culling, and maybe threats to entire containers or storage unit. So because these diseases can create defect and these defects can result into economic loss, the individual defective fruit is handled additionally, like for example culling means removing the specific part which is diseased. Now, nest production can be done to restrain or refrain product to product spread with visible pathogen development. Now, what is nest production? Nest production means the cutting of fruits and vegetable in a particular manner to decrease the waste and to avoid the disease. The parts which can have higher microbial load and can create defects, and without them the product can be, fruits or vegetable can be marketed or transported. The nest production is done there. Then spread of decay within container by fluids produced during disease development can also cause problem. So culling can be effective in this case. Volatiles produced during infection lead to off flavors, undesirable aroma, hypoxia, and even mycotoxin patulin generation. Now, volatiles produced during infection may lead to off flavors and undesirable aroma, and because of high concentration of these off flavors and undesirable aromas, hypoxia, the reduction in the oxygen concentration can be there. And then mycotoxin. What is mycotoxin? Mycotoxin is a kind of toxin, and patulin toxin is not destroyed by pasteurization or thermal denaturation. So vigorous heat treatment cannot degrade or destroy the mycotoxin patulin. So it is very much important to remove all these defective parts or defective fruits which have disease. Now, concepts important in post-harvest decays. First is severity. What is severity? Amount of host tissues involved. Percentage of host tissue. What is host tissue? The fruit or vegetable tissue involved in the disease, or it is infected by the disease. It is termed as severity. Now, the incidence, the percentage of product with lesions. Lesions means the ghav. So percentage of product with lesions or wounds is known as incidence, percentage of incidence. Now, inoculation. Pathogen becomes located in an infection coat where it can infect plant. So when pathogens are in situation to infect plant, this situation is known as inoculation situation. Then penetration, actively or passively penetrate outer barriers such as cuticle, bark, etc. So penetration means coming of infection inside the fruit or vegetable. Now, infection, pathogen begins to colonize, means pathogens, there's disease causing microorganisms begin to multiply within the living plant tissue is called infection. Then latent period between arrival on the plant to shedding of pathogen structures, that is secondary inoculum. Latent period means the time between arrival of pathogen on the fruit and development, start of the development of multiplication. In between the period is called latent period. Latent heat we have studied in other subjects, that the heat which is used for the phase transfer. So same is like that. When pathogen arrives on the plant and when it start multiplicating, so it changes its phase. So in between the period of two phases is called latent period. Now incubation period, the inoculation to symptom development. Incubation period is when you when anyone is in the position to know that the disease has been developed, is the incubation period. So maybe pathogens start multiplicating, but there will be no symptom. So symptom development, arrival on the plant, disease pathogen arrival on the plant to symptom development is the incubation period. Now quiescent, pathogen initiates infection and then stops development temporarily. This quiescent situation is when pathogen initiates infection, means it multiplies heavily, increases heavily, then the plant system, the fruit and vegetable system, stops its development and starts targeting the infection or to combat infection. This stage is known as quiescent. So at the latent phase, infection is not visible. Now signs, visible symptoms, visible pathogen structures are sign. Symptoms are visible changes in the plant. So there is difference between signs and symptoms. Signs are visible pathogen structure. We can identify that this is the microbial development, and the symptoms are visible changes like sogginess, like wounds, etc. Now what favors post harvest disease? This is disease triangle. It is not clear here. We can see it clearly here. So this is the disease triangle. You can see the three legs. What are the three legs of triangle? First is susceptible host. Then second leg is conducive environment, the favorable environment, and third is the pathogen. Or we can say the triangle starts from first leg, that is pathogen. Pathogen should be there for having disease. Then second leg that should be there, that is susceptible host, which can give environment to the pathogen to multiply. So, and the conducive environment, the environment outside the host which supports the pathogen, or inside environment that can support the pathogen. And susceptible host can be mechanically damaged, physiologically damaged, or lower in nutrient. Whatever the condition, susceptible host can be like that. So all three legs, first leg pathogen, second leg susceptible host, third leg conducive or favorable environment, is necessary to initiate disease. So this is the disease triangle. So technically or scientifically, if one leg of the triangle is absent, disease does not occur. Now, what are favorable or conducive environments? That are wet or humid condition. That canopy means the shade where fruit or vegetables are kept is wet, or soil or beds, maybe flooding. Dew, that is condensation water and guttation are slow to dry in the morning, so availability of moisture on the plant or fruit. Then prolonged periods of wet canopies, so there is more rain. Then favorable temperature, so it depends on the individual crop and pathogen, and not exposed to UV or desiccation, means sun or the dry condition. Desiccation is the dry condition. Now moving to the large populations of pathogens. Freshly harvested fruits and vegetables are not sterile because they have microbial load due to environment, soil, or much factors are there. Much of the naturally occurring biome, that is microflora, flora and fauna, can produce a decay if the product is abused, means if the product is open to the pathogens. Debris from previous crops, senescing means leaves which are dead, floral petals which are dead. So the debris of this can be the home for these pathogens, and these will increase the multiplication of pathogens. Runoff from rainfall, including temporary flooding, then cankers, diseased or dead branches, survival structures on or under trees like sclerotia, mummies, pre-harvest diseases on foliage. So anything like that. Sclerotia is the multiple hyphae, the bunch of fungal hyphae structure which can be dormant for long time. So spory structures and all the things which can support the growth of pathogens. Then equipment and facilities, the field bins, picking bins, the storage room, coils which are not clean or sanitized time to time. Then waste disposal sites, the dump, cell product dumped near production sites. So these can initiate the pathogen, because these are the large population of pathogens, and from this fruits and vegetables can be infected. So these are also precautions to take. Then series of inoculum. We know what are the sites of large pathogen, then how these can be inoculated. As we know, these are generally ubiquitous, they can be found anywhere. Then debris, of course, we know. Then weeds, then they can survive during the wet conditions, so favorable conditions. Now moving towards the survival in sclerotia. So we can see that this is fungus sclerotia. These are hard, dark, rusting body of certain fungi consisting of a mass of hyphal threads capable of remaining dormant for long periods. Now moving to the dispersal from survival structure or during secondary spread in crop. Lightweight or relatively dry spores can be dispersed by wind, so inoculation can be by dispersion through wind, through rainfalls, or through storms, or through flood. So we know where large number of pathogens can be found and how their inoculation into the host plant can be occurred. Now the susceptible plant and the third leg of the triangle. So what is susceptibility? Normal susceptibility is certain cultivars may have levels or resistance. So resistant variety have normal susceptibility. Then physical injury, physiological injury, and disturbance into ripening or senescence process can make susceptible the plant susceptible to the fruits or vegetable susceptible to the pathogens. Like physical injury is there, so pathogens can grow. Then physiological injury like nutrient deficiency, pollination issue, or uneven moisture content, so these can trigger the pathogen growth or inoculation. Now types of decay. So there are soft rot, second is grey mold rot or sore rot, and third is rhizopus rot or mucor rot. So then necrotic spot, then after that alternaria rot or cladosporium rot. Soft rots are softened with general breakdown of structure. The appearance will be soft, so these are called soft rot, and it will be because of the general breakdown of the structure. Then grey mold rot means the soft rot plus sporulation of specific variety. So it can be grey mold rot or sore rot. Then soft rot with extensive mycelial growth like cottony appearance. These are rhizopus rot or mucor rot. Then necrotic target spots, their dead cells are there, necrotic, and sporulation will be there, so it will look like a firm dead structure. Then firm dead structure covered with the mold is alternaria rot or cladosporium rot. Then bacterial soft rot exists as epiphytes or saprophytes. Now, what are epiphytes? Epiphytes is an organism that grows on surface of a plant and derives its moisture and nutrients from the air, rain, water, or from debris accumulating around it. Epiphytes takes part in nutrient cycles and add to both the diversity and biomass of the ecosystem in which they occur like any other organism. So these are epiphytes who live on the organism, but they work for nutrition Then saprophytes, a plant fungus or microorganism that lives on dead or decaying organic matter. So eating dead is saprophytes, and epiphytes live on living organisms and take part in nutritional cycle. Then rainfall, irrigation, water, insects, machinery, field operations such as pruning, so move bacteria to the plant. We know all that bacteria may survive and multiply on plant's surface if wounds or free moisture will be there. So intercellular spaces of plants are ideal moist chambers because intercellular spaces have free water, which bacteria use as a carrier or transportation vehicle to have nutrient from cells and to move their metabolic products towards the fruit or vegetable cell structure. So bacteria produce extracellular enzymes that attack pectolytic compounds. Pectin compounds are what? They give structure to they are the fibrous part of the carbohydrate, and they give structure to the fruits and vegetables. So these bacteria produce extracellular enzymes that attack pectolytic enzymes compounds that glue cells together or produce lactic acid that kills cells. So basically metabolic product of the bacterial digestion will kill the cells of the fruits. Now you can see the soft rot in the broccoli and the tomato. Now another bacterial soft rot in the tomato. Now to know the gravity of or extent of infection, how much percentage of infection is how much percentage of infection spread is there, the internalization of bacteria modeled in experiments with India ink. India ink is a suspension of carbon particles that are of size of gram-negative bacteria. So India ink has been used to study the sutures on cadaver eyes. If the ink particles penetrate, so can bacterial pathogen. Now, what are sutures? Sutures are junction of contagious plant parts. So, and the cadaver eyes are the toxin producing parts of the diseased bacteria. So the juncture of these, at this juncture, if it is showing on the upper surface, the India ink is penetrated, the suspension of India ink is penetrated towards, and we can see that India ink has internalized, and we can see the how much infection is spreaded here in the tomato. So to know the extent of infection, the India ink internalization is used. So what are the factors that can affect the internalization of ink. That is, the first is temperature. If temperature decreases in tissue, it creates vacuum in the intercellular spaces. As we know that when temperature increases, the amount of oxygen ingestion is more and metabolic activity is more, and when metabolic activity is more, then heat generation will be more. If temperature decreases, the heat generation will be less, and when heat generation will be less, then evaporation of moisture within the cell system will be less, and there will be low vapor pressure in the internal system or the intercellular spaces. If water covers pores between fruit surface and external environment, vacuum draws water into the pores. Note: tissue cooling and water uptake is an issue associated with hydrocooling. So, if water is there on the surface, it can be moved towards the internal system and it can create the environment which is favorable for the pathogen growth. So the pressure of water is also important. Then we were discussing the sore rot, and you can see the sore rot, how it will look like. In sore rot, lactic acid aroma will be present. Now we have seen the third type of rhizopus, third type of soft rods, that are rhizopus rod. You can see the white rod, that is sclerotia, the fungus, the cottony appearance, cottony and fuzzy appearance, then brown rot of stone fruits. So rhizopus rot in the tomato. Then internal alternaria, where general appearance of several firm decays of tomato fruit, and from the upper part they are firm but they are infected. So internal development can be there. Then penicillium molds, you can see and recognize the penicillium molds infection. Then botrytis fruit rot, you can see and recognize the botrytis fruit rot. This is the anthracnose, the necrosis, the firm necrosis, you can see and recognize. Then you can see the brown rot and causative agent, the causative pathogen you are seeing in the slides. Then certain pre-harvest diseases lead to post-harvest problem of a non-infectious nature. They don't spread into product to product, means from one product to another product they don't spread, but these are the pre-harvest diseases. These are individual infections. You can see the viral diseases, internal necrosis in the tomato, where you can see the almost absence of pulp. Then bacterial blotch, Blotch means dag. Some of watermelon. So these are the pre-harvest diseases which are non-infectious in nature. They doesn't spread from one to another. So I think the diseases, the post-harvest diseases are clear. So if you have any query, you can post in the comment box or you can contact me. Prepare handwritten notes from the lecture.